Hello, everyone. Welcome to part three of the reproductive system. Today, we're going to start our discussion of the male reproductive system. So like the female system, we're going to split this into two basic parts. So here in the male anatomy, the primary sex organ are the testes. This is what is going to be producing gametes and producing hormones to help regulate the reproductive or male reproductive system. From the testes, we're going to have a series of tubes and structures designed to deliver the gametes where they need to be, right, inside the female reproductive system. Just a point of clarification, the penis is the copulatory organ, the organ that you're going to have sex with, but it is not the primary organ, not the primary reproductive organ. So just like we saw with the female anatomy, make sure you get those two things kind of clear and separate. Here we have our gross mid-sagittal cut through the male reproductive system. We have the urinary bladder, we have the rectum leading to the anus. So that helps us get our bearings, right? The penis is fairly obvious. We have the testes down here in the scrotum. Here we have some structures you may be a little less familiar with. Over here on the posterior side of the testis, we have something called the epididymis. The epididymis is going to connect testis, so for singular, one of them, right, to what is called the ductus deferens, or perhaps more commonly known as the vas deferens, right? This is the structure that connects the testis with the prostate. So this structure serves a solely reproductive purpose. Here, we have the testis to the epididymis to the ductus deferens. Notice that the ductus deferens is eventually going to widen to a structure called the ampulla. And then the lumen of the ampulla is going to open into the prostate gland. Another nearby organ called the seminal vesicle also joins the structure. We'll talk about those later. Here in the bladder, right, we have the prostatic urethra, which will then meet the opening of the ductus deferens, right? That space, that particular space, is called the ejaculatory duct. So, I'm getting a little ahead of myself, but let me just clarify this to make life easy. Testes connect to the epididymis. The epididymis connects to the ductus deferens. Here, the ductus deferens widens into the ampulla. Right there, it will then join with a duct draining out of the seminal vesicle. The opening of the seminal vesicle and the opening of the ampulla of the ductus deferens, these two spaces join together to form a structure called the ejaculatory duct. The ejaculatory duct then meets and unites with the prostatic urethra. Right now, the rest of the system is going to be shared between urinary and reproductive. So this was a fundamental difference. Remember, in the ladies, urinary and reproductive was totally separate. Here, urinary includes bladder, prostatic urethra, membranous urethra, and then spongy urethra. Reproductive, right, production of sperm is going to happen in the testis to the epididymis to the ductus deferens to the ampulla, and then you're going to have glands add other fluid that we'll talk about later. Right, these structures so far up to the ejaculatory duct are just part of the reproductive system, right? So when that prostatic urethra joins with the ejaculatory duct, then we start the sharing of the reproductive and urinary system in men. Right. Oh, I should point out here, notice this tube going up, this is one of the ureters, right? So this would be the left ureter going up to the left kidney. Let's see, we can see about a third or more of the penis is actually inside the body, and it is actually anchored to the pubic bone. As we mentioned before, the tip of the penis is called the glans, just like the tip of the clitoris was called the glans. And we're going to have, not labeled here, but we're going to have another fold of skin on top of that, the foreskin, which is also called prepus, right? Or foreskin right here. All right, I do believe that is all our major structures we need to talk about. So scrotum, right, is a skin covering that covers the testes. right? This is something that I alluded to earlier, right? Why is there a scrotum, right? Why are the testes outside the abdominal cavity? That's all really weird. And the answer has to do with temperature, right? You get optimum sperm production at about 3.5 degrees below body temperature, right? That is why the testes are on the outside of the body, so they can be cool enough to make healthy and efficient sperm. That's the whole reason why when they're created up in the abdominal cavity during fetal development, before we're born, before men are born, right, the testes are going to descend out the abdominal pelvic cavity. They're going to enter into the scrotum, and that's how that occurs. Remember from the very beginning of lecture that the scrotum is homologous to the labia majora in females. So during that fetal development, a structure that would become the labia majora in women will actually join together to form the scrotum in men. Now, there is a line that runs in the mid-sagittal direction separating the two halves of the scrotum, and that's called the raphe. So the raphe is just that line separating the right and left sides of the scrotum. In the wall of the scrotum, we have a layer of smooth muscle called dartos muscle. So we have this condition whereby we need to keep the testes about 3 degrees below body temperature, so they're outside of the body. Some of you may have noticed that you can have varying levels of distension of the scrotum from the body wall. Part of that is controlled by the dartos muscle. Dartos muscle is smooth muscle, so you do not have conscious control over this muscle. So one of the functions of having this muscle is if it's real cold outside, you're going to get shrinkage. Why? Because that is to keep the testes closer to the body to help warm them up. Because if they get too cold, that's no good, right? So you go out in the Pacific Ocean, get in an Alaskan current, and it gets real cold, whoop, you're going to get some shrinkage going on to try to maintain that optimum temperature. Likewise, if it's a really hot day or your body temperature soars, you're going to get distension because they're trying to get greater distance. The dartos muscle is trying to increase the distance of the testes from the body in an attempt to lose heat, in an attempt to keep this optimum temperature, about 3 degrees below body temperature. So it's a thing, fellas, if you're trying to be a dad or you're trying to conceive, boxers and not briefs, it will definitely actually have an effect. So just a little bit of information for you. Okay, so let's take a look at the structure of the scrotum. So here we have the right and left halves of the scrotum. Here's that raphe that separates right and left halves. Deep to that, we have the dartos muscle. Now, deep to that, we're going to have the external spermatic fascia. So this is a layer of connective tissue just inside, right? Or actually, I should say, inside the dartos muscle, we have a structure called the spermatic cord. So the spermatic cord, it has several layers to it. The first outermost layer of the spermatic cord is the external spermatic fascia, that layer of connective tissue that I just talked about. On this image here, if you cut open the external spermatic fascia, you will see muscle. Right, you will see the cremaster muscle. So the cremaster muscle is another muscle. This one is inside the spermatic cord. You have one spermatic cord for each testis. So you've got one dartos muscle in the scrotum, but you have two cremaster muscles, one for each spermatic cord, one for each testis. Now, where does this come from? Remember, in fetal development, the testes start inside the abdominal cavity and they actually descend out the abdominal cavity. They actually have to pass through the abdominal muscles. So the cremaster muscle is actually derived from your abdominal muscles. Your abdominal muscles are skeletal muscles. So the cremaster muscle is skeletal muscle. You could, at least in theory, have some conscious control over that. So there is our cremaster muscle. If you open up the cremaster muscle, right, you're going to find the internal spermatic fascia. If you cut open the internal spermatic fascia, now we've gone all the way through the layers of the spermatic cord. What you're going to find in there is the testis itself, Sitting on top of the testis is the epididymis. You're going to find the testicular nerve because there's going to be, important autonomic nerve functions going through here, and sensory nervous information is going to be passing through here. We're going to find the testicular artery, if that makes sense. But we have this other interesting structure here called the Piniform Plexus. So this big convoluted spider webby looking vein thing, the Piniform Plexus, and you might look at that and go, what's up with that? And actually, believe it or not, that actually acts like a radiator to help dissipate heat. Remember, the testes need to be kept below body temperature. And one of the functions of the Piniform Plexus is to increase surface area and actually dissipate heat and act as a cooling system for the testes. So interesting little structural function there. Oh, and remember, right, the testicular artery, testicular vein, right? Remember when we looked at the gonadal artery and vein, right? They're way up by the kidneys, right? And it makes sense for the ovaries because the ovaries are right there, right, in the abdominal cavity. But in men, right, this vessel still starts way high up superior and it just travels all that long distance to leave the body. That's just like another little bit of evidence for the migration of the testes out of the abdominal cavity down into the scrotum. I do believe that's all the structures that we need to talk about, right? This is what it looks like in an actual individual. You can see the spermatic cord opened up, testis and epididymis. So just going over in words what we already talked about, the spermatic cord originates from the inguinal canal. That's this space right here. This is that inguinal ligament running from the iliac bone to the pubic bone. The opening is just where the spermatic cord is going to pass through, right? Inside the spermatic cord, you're going to find the testicular artery, which is a branch off the abdominal artery. The Piniform Plexus is the network of veins that helps with cooling. Cremaster muscle is going to be formed by the internal oblique abdominal muscles. And you're going to have various autonomic and sensory nerves that need to innervate the testes. One thing that I should also point out, just as an interesting fun fact, when the testicles are damaged or abused, right, it is extremely painful, and it's a particular kind of pain, right? It's a visceral pain, right? Because these are organs which are normally supposed to be inside your abdominal cavity are now outside your body, right? Because they're outside of your body, when they get injured or, they get damaged, it's going to hurt like somebody is just, kicked you in the spleen or something like that. So anyone who's had that experience, this is a little bit of an explanation why, right? Because these are organs that are normally part of what derived from or created from visceral organs, right? Or in that same area. Okay. So the particulars of the testes themselves, right? Each one is an oval organ inside the spermatic cord, inside the scrotum, right? Their main function is the production of sperm, which are the gametes, right? Carry half the genetic information that the father is going to donate. And the testes will produce androgens, which are male hormones. So the most well-known example of this would be testosterone. Now here's a confusing one. Each testis is covered anteriorly and laterally, so on the front and the sides, by a serous membrane, okay, called the tunica vaginalis. So we have the primary male sexual organ covered by a serous membrane called the tunica vaginalis. Ugh. What's up with that? Okay, so the story is the root vagina or vaginalis means ensheathing, right? Like you put a knife inside a sheath, that's just what that term comes from, right? So this is a structure that ensheathes deeper parts of or covers other parts of the testis. I'm sure that you can have a humanities class that can write a whole dissertation just on this naming scheme right here. I am not commenting on any of that. I'm just trying to explain these are the anatomical terms, and this is where they come from in an attempt to help make some sense to you guys. I didn't come up with this. But anyway, here you go. primary male sexual organ is partially covered, front and sides, anterior and lateral, by a serous membrane called tunica vaginalis. So just like any other kind of serous membrane, we're going to have a parietal layer, we're going to have a visceral layer. Let's go to the picture. So this pink thing right here, it's perfect color choice, right? This pink thing right here, that is the tunica vaginalis. Right? It has a parietal layer and it has a visceral layer in contact with the deeper part of the testis. Now, what is this white area? This is the Tunica Albugenia. So remember, we also had a Tunica Albugenia when dealing with the ovary. So if you remember, 2 lectures ago, right, the structure of the ovary, we had the germinal epithelium, and deep below that, we had Tunica Albugenia. Well, here we have a Tunica Albugenia in the testis. So this is really a layer of connective tissue, which is going to form kind of the body or the structure of the testis. The material of the Tunica Albugenia will dive down and subdivide the internal space of the testis. Those subdivisions are called septum. So the septum will define spaces with tiny little tubules where the sperm are actually produced. So that structure of all those tubules is called a lobule. So that is a lobule. These little tiny tubes in here, these are the seminiferous tubules. So if you take a cross section of the seminiferous tubule, this is the tubule itself. Out here on the outside, you're going to have a combination of what are called sustentacular cells, also called nurse cells. These cells aid in the production of the sperm. Out here in the periphery, you're going to get, you will find immature spermatids, and as they mature, they're going to move closer and closer to the center of the tubule. When they reach the center of the tubule, when they're mostly mature, then they can be carried away where they can be used. As you see, the further and further you get closer to the center, the more sperm or tadpole-like these cells get, right? So these really are starting to develop their flagella, whereas out here, right, not so much with flagella. Outside the seminiferous tubules, you'll have what are called interstitial, because they're in between one seminiferous tubule and another. These interstitial cells are the cells that are going to be producing the androgens, like testosterone. Okay, so let's get back to the larger structure of the testis. We have the seminiferous tubules, all packed in one little space is a lobule. One lobule is separated from another lobule by the septum. All these seminiferous tubules are going to lead out of the lobules to a space here, space called the mediastinum of the testis, right? So we had a mediastinum in the chest. We also have a mediastinum in the testis. So the mediastinum of the testis is a space. The tube, right, that is passing through this area is called the reprotestis. So seminiferous tubules connect to the reprotestis. The reprotestis connects to the epididymis by the efferent ductals. Efferent ductals. So the epididymis has a head, it has a body, it has a tail. The tail of the epididymis is continuous with the ductus deferens or vas deferens. So you're going to want to be able to put together all these structures and know the path of gametes out of the male anatomy. It will start in the seminiferous tubules. They will pass to the reprotestis, from the reprotestis to the efferent ductals, to the head, body, and tail of the epididymis, to the ductus deferens. From the ductus deferens, right, you're going to meet, it's going to turn into the ejaculatory duct, which it will join with the prostatic urethra, to the membranous urethra, to the spongy urethra, out the external urethral orifice. That will be the flow of sperm created in the testis and eventually leaving the body. Any major structures that I have left out? No, I think we've covered pretty much everything here. Just a little factoid, a vasectomy surgery involves making incision in the lateral side of the scrotum, and they took something that looks very much like a knitting needle, and they reach into the spermatic cord and they start pulling out structures. And when they pull out the ductus deferens, They just give it a little snip and they cauterize it, and they just tuck those two now severed ends of the ductus deferens back inside the scrotum, and you're snipped, right? Sperm no longer have a pathway to get from the testis out the rest of the reproductive system. Now, some people ask a question, well, what's going to happen with all the sperm if they can't get out, right? Is it going to be a problem? It's not a problem. Sperm are normally stored. The sperm that are going to be used, right, for possible conceiving a child or for copulation, right, the sperm are actually stored here in the epididymis. So in the testes, this is for the long-term development of the sperm. When they're more or less ready to do their thing, they're going to be transported and stored in the epididymis. And then when you have intercourse and ejaculation, the sperm are released, then they're going to become active, then they're going to start swimming. But if they're not used, when they're just hanging out in the epididymis, if they get old, they start breaking down, they're simply reabsorbed by the body. They simply go away. So it's not an issue that they can't get out of the scrotum. So they just simply get reabsorbed by the body. It's no big thing. Now, one little factoid, early on when these surgeries were really sort of first being produced, like they're first being done on a large scale in like the 80s, or at least I know in the 80s, they actually did not cauterize the ends of the ductus deferens. And I actually knew an individual who had been circumcised, and lo and behold, 15 years later, his girlfriend at the time said, "Guess what? I'm pregnant." And it just so happened that those two ends of the ductus deferens found each other and healed, and he had a spontaneous reversion of his vasectomy. Nowadays, when they pull those ends out, they cauterize them or burn the ends to make that much less likely. But it can happen. Okay. So let's go over in words some of the structures we just talked about, right? Testes located deep to the visceral layer of that tunica vaginalis, right? That layer that the visceral layer of the tunica vaginalis are covering is called the tunica albugenia, right? The tunica albugenia is going to, right, project to the interior of the testes and create that space, the mediastinum of the testis. Through the mediastinum of the testis, that's how blood vessels and nerves are going to get into the testis, and how the retrotestis is going to carry sperm out of the testes. So each testis is subdivided by structures called septa. Septa create lobules. There's about 200 to 250 lobules per testis. Right. Inside the lobule, you will find seminiferous tubules where you will find the cells for the creation of sperm. So here in the seminiferous tubules, you're going to find inside the tubules, you're going to find two different types of cells. The nurse cells, also called sustentacular cells, non-dividing cells that will help in sperm development. They will also form the blood-testis barrier. So we had a blood-brain barrier, but now we have here a blood-testis barrier because the sperm are very, very important, right? You don't want toxins or unwanted compounds getting into these rapidly dividing cells and scrambling the genetic material you're trying to pass down to the next generation. So there's an extra layer of protection here to prevent the sperm from being damaged by any environmental toxins, because that's, you know, from an evolutionary point of view, this is why dudes exist, right? To pass on their genes, right? So you want to pass on your genes accurately. You don't want to pass on kind of junky genes that got messed up because of some environmental toxin. You want to keep those things safe. So we have this blood-testis barrier. And of course, you're going to have the cells that will eventually turn into sperm themselves. Right. Outside of the seminiferous tubules, you'll have the interstitial cells, which I pointed out earlier, and their job is to do things like produce hormones like testosterone. Right? A male hormone is simply called an androgen. So those are the interstitial cells outside the seminiferous tubule. The big cells here, those are the sustentacular ones, the little tadpole guys. Those are your developing spermatids. So a little cartoon picture, right outside the seminiferous tubule. Those are your interstitial cells. These big, giant guys, a little hard to see in the histological slide, but the cartoon slide makes it real obvious. These are the sustentacular cells. These are the nurse cells that aid in the development of the spermatids. Over here, you're going to have the stem cell giving rise to spermatids. Creation of sperm is called spermatogenesis. It's also a form of meiosis, right? We have to go from 46 chromosomes down to 23, right? So we're going to have to lose half of that genetic material. So again, I'm not going to test you on the details of spermatogenesis. This is not a class where I'm going to teach meiosis. But if you want to know, you can read this table and check it out, right? 46 pairs, or I should say 46 chromosomes. Eventually, we need to get down to 23. Eggs got 23, sperm's got 23. Put these together, you got 46, and now you have the blueprint for the next generation. Right. This just gives a little bit more detail going from a stem cell down to a sperm itself. Right. You have three basic pieces of the sperm. You have the head, mid-piece, and tail. The head, this is going to contain the actual genetic contribution of the father. Right. His 23 chromosomes are going to be tightly packed away inside that head, and it's covered by this acrosome cap. Basically, it's got a little cap on it with digestive enzymes. The sperm is going to use these digestive enzymes to burrow its way to the ova, right? Once that sperm passes the zona pellucida, you're going to have a rapid change in the zona pellucida, and all other sperm are going to be locked out. One sperm in, then the egg sort of drops the gate, locks the doors, because you don't want any other sperm getting in, because you only want 46 chromosomes. If you ended up with 69 because you got 2 sperm in there, right, that would be a problem, right? That egg would never develop, right? That embryo would never develop. So the mid piece connects the head to the tail. The tail is basically just a little propeller thing, the flagella. The mid piece, this section here actually has a large number of mitochondria. So this is going to be, and they're actually special mitochondria because sperm needs a lot of energy in order to work that flagellar tail. So this is where you're going to find a whole bunch of highly organized and kind of unique and interesting mitochondria, but we're not really going to discuss that here. Think of this as the engine, the outboard motor for the sperm, right? And then the tail would be like the propeller of a boat. So if you want to know a little bit more detail about sperm development, here you go. I'm not going to test you guys on it. And that brings us to the end of part 3, right? Covering the primary sexual organ in men, the testis. Next time, we'll start talking about the rest of the accessory organs to help deliver the sperm to where they need to be. See you next time.